my dear students, I hope you are all have been doing well. We are going to study about amino acid catabolism. This is my lecture number 12, and in this lecture we would be studying that how amino acid within our cell could be catabolized, means could be further metabolized. So we all know about this fact that amino acids are the building block of proteins, and whatever from where these amino acid comes in our body, that they can be already synthesized in our body, and they can be present in our food, so we can take them from outside our body in the form of proteins. So proteins are the macromolecule that are made up from the amino acid. So thinking about this fact that if we are having protein in our diet, and this protein is having long polypeptide chain, so first process for the the first process would be the breakdown of the protein into small pieces. So this process is known as the degradation of protein or the digestion of protein. In that, protein long polypeptide chain would be converted into small pieces, or would be chopped out into small pieces by the by the mechanism of proteolysis. And then this mechanism would be takes place with the help of an enzyme that are called as proteases enzyme. So these proteases enzyme, actually these are the proteolytic enzyme that chop the peptide bond or break the peptide bond between the two amino acid, or within the that are present in a polypeptide chain. So these protease enzyme, actually these are the hydrolytic enzyme that break down the peptide bond after the addition of a molecule of water. As the name of these enzyme are indicating that these are the hydrolytic enzyme. So how would they perform their mechanism of hydrolysis? They will add a water molecule in the bond, and in the peptide bond, and then eventually the peptide bond would be break down. So you know what, you all know that what is the peptide bond. Peptide bond is a bond that join two amino acid together, and eventually they when more than a number of amino acid would be join together through peptide bond, so they will make a protein polypeptide chain. So this process of proteolysis or hydrolysis, so hydrolysis of peptide bond would takes place in the proteasome. That is an organelle present in our, that is also a protein. Proteasome is also a protein that is having the the that can be perform the mechanism of proteolysis. Proteolysis means the hydrolysis of protein. So the hydrolysis of protein would be takes place in the within another protein, that is a big protein, and this is a cylinder shaped protein, and we name this protein as a proteasome. So the small protein or small polypeptide chain will come and inside this proteasome, where this hydrolytic cleavage of the peptide bond would be takes place, and then after the hydrolytic cleavage, these long polypeptide chain would be converted into small protein. So and then for this purpose, the enzyme different enzyme would take part, and these can be serine, aspartate, or metalloproteases. And one of the serine protease that we have already studied in our lecture number 10, that is the trypsin. It is one of the proteolytic enzyme, or it is one of the protease enzyme that break the peptide bond. So what happened, all this happened in our the breakdown of peptide, the breakdown of the peptide bond, and uh, breakdown of the peptide long polypeptide chain into the dipeptide and triapeptide would be takes place in the in our means in the organ of our body. And what are these organs? For example, these are the intestine and the stomach. However, what happened after that, the amino acid, after the cleavage of the peptide bond, amino acid would be released from the polypeptide chain, and the single amino acid would be enter inside the cell. So after the entry of the amino acid inside the cell, the amino acid would breakdown would be start. So the amino acid breakdown, uh, from where it will be start? It will be start when the amino acid would be released from the long polypeptide chain after the digestion in the digestive tract or digestive organs. And then when the small amino means individual amino acid would be enter inside the cell. So the amino acid breakdown or amino acid catabolism would be start inside the cell. And the process of amino acid, what is the process by which amino acid further catabolize or further means metabolize? There are two means mechanism of amino acid breakdown within the cell. One is the oxidative deamination, and one is the transamination. So the oxidative deamination is a process, or is, as the name of this mechanism indicated, this is a method, or this is a mechanism by which um, amino group from the amino acid would remove by the action of the enzyme deaminases. So deaminases are the enzyme that are present within our cell. What does this enzyme do? These enzyme remove the amino group from the amino acid. And what happened then? This amino group would be converted into ammonia, and cannot and then this ammonium will enter in the urea cycle with and this takes a place the in urea cycle would takes place in our liver. So what happened, they within our cell, what happened, the amino group from the amino acid would be removed by the enzyme deaminases. And then what happened, this removed amino group would be converted into ammonium, and then ammonium will be enter in the urea cycle. And after the entry of the ammonium in the urea cycle, the urea cycle convert this ammonium into urea, and then urea would be released from our body through the urine. So why this means this long um, uh, question is why this long mechanism is taking place in our body cell. So why this amino group is converting into ammonium, and then ammonium is converting into urea, and then urea is releasing from our body, because why amino group that is converted to ammonium could not be removed from our body in the form of ammonium. The reason is that ammonium is highly toxic in nature. So for this reason, the body liver our liver in the body, a cycle takes place, that is the urea cycle. So our body would convert this ammonium that is highly toxic in nature. uh, 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 the liver would convert the ammonium that is highly toxic in nature into urea. So the urea is less toxic than ammonium, then urea would be removed out from the body um, in the less toxic form. So what happened after the oxidative deamination? We were discussing about the oxidative deamination. So oxidative deamination is the process of the breakdown of amino acid. In by this process, this is the first step of amino acid breakdown. And in this step, what happened first? whatever amino acid we would be having in our body cell, in the first step amino group of this amino acid would be released and by the action of enzyme deaminases, and then this amino acid would be the amino acid would be means convert into ammonium, and ammonium would be enter in the urea cycle and convert into urea and excrete out from our body. However, the remaining part of the amino acid, what would be the remaining part of amino acid? It would be having the carboxylic group, an R chain, and then a hydrogen atom. If you remember, I talk about the structure of the amino acid in very first previous lecture, lecture number 10, that amino acid consists on four basic part. One is carboxylic group, one is amino group, one is R chain, and one is hydrogen atom. So after the removal of the um, amino group from the amino acid, three part would be remaining in the amino acid molecule, that would be the carboxyl group and the R chain and the hydrogen atom. So this remaining part, after the removal of ammonia from the amino acid, after the removal of amino group from the amino acid, the remaining part, what would be having with the remaining part, then of the amino acid, the remaining part of the amino acid would be further oxidized and form alpha keto acid. And then what would be happen with this alpha keto acid? The alpha keto acid would be enter into the TCS cycle and and recycle to produce energy, and then it can be enter this alpha keto acid can be enter in the glycolysis and convert into pyruvate, or it can be a pyruvate, and the pyruvate can convert it into acetyl CoA, then acetyl CoA further recycle and form energy. So we can easily understand this all means picture within this diagram. So what happened, just if assuming that we were be having an amino acid, any amino acid, and this amino acid would be having four part. One was the carboxylic group, one was the amino group, one was the hydrogen atom, and one was the um, uh, carboxylic group, amino group, hydrogen atom, and one was the R chain. So in the first step of the amino acid catabolism, amino group removed from the amino acid, and then remaining amino acid enter amino group removed from the amino acid, and that amino group enter in the urea cycle in the form of ammonia and excrete from the body. However, the remaining part of amino acid convert into alpha keto acid. So this is the one of the example of alpha keto acid is the alpha ketoglutarate. This is the alpha keto acid. So this alpha keto acid then enter in the TCA cycle, and then going through all these reaction and convert into oxaloacetate. Then this oxaloacetate can be convert into phosphoenol pyruvate, and this phosphoenol pyruvate means can be further converted into pyruvate, and this pyruvate is can be decarboxylate and then converted into acetyl CoA. Then this acetyl CoA then further proceed through this TCA cycle and form energy. So this was whole the process means how the amino acid means further catabolize and produce energy by oxidative deamination. However, amino acid can be amino acid can be further catabolized by the mechanism of transamination. And what is the transamination? Transamination reaction is a reaction by which the amino group from an amino acid transfer uh, um, into another alpha keto acid, and then the alpha keto acid would enter again this TCA cycle, and then similarly recycle as the amino acid recycle after the oxidative deamination of the of the amino acid. So the what is the difference between transamination and the deamination? In deamination reaction, what happened? Uh, Amino group of an amino acid would be lost. How it would be lost? Would be lost after the conversion of amino group into the ammonium, and then ammonium would be enter in the urea cycle, and then urea cycle convert this ammonium into urea, and then it will be lost. However, in the transamination reaction, what happened? The amino group from an amino acid would not be lost. It would transfer from an amino acid into the alpha keto acid, and then this alpha keto acid would be further means metabolized through TCA cycle, and then it could be convert into the pyruvate after going through the glycolysis cycle. So for the transamination reaction, the enzyme that are used for the transamination reaction are the enzyme we call them as transaminases. And for the deamination of the amino acid, so the enzyme that would be take part for the deamination are the deaminases. So the transamination and deamination are two mechanisms that are important for the catabolism of amino acid. But for what the means mechanism amino acid would be adopt for their catabolism, either they adopt the transamination reaction, or they adopt the mechanism of deamination for their metabolism. After adopting this mechanism, either transamination or deamination, what would be happening? The product of transamination and deamination reaction would be enter into the TCA cycle in the form of alpha glutarate, and would be converted into then succinyl CoA. Then succinyl CoA would be further converted into succinate, and then fumarate, and then malate, and then oxaloacetate, and then it can be further recycled through this TCA cycle, or it can be enter again into the means glycolysis cycle after conversion into phosphoenol pyruvate. Here we can see that this phosphoenol pyruvate convert into glucose, and then this glucose will enter into the glycolysis pathway and break down into two molecule of pyruvate and produce energy in the form of ATP. So the end product of amino acid breakdown would be the production of energy in the form of ATP. So the whatever the process of their catabolism, amino acid would be adopt, either it would be deamination or oxidative deamination, or either it could be transamination. Eventually amino acid would be end all those amino acid that that are adopting the process of either transamination or deamination for their metabolism would be eventually end for the production of energy in the form of ATP. And how it could be happen? It could be happen because after the removal of ammonia from uh, after the removal of amino group from the amino acid, they would convert into alpha keto acid, and then alpha keto acid would be enter in the TCA cycle, or further metabolize, and then it can be enter in glycolysis and glycolysis pathway and further metabolize and produce ATP. However, we have to keep in mind one thing, that after entry of this alpha keto acid in the TCA cycle, these amino acid that start means catabolizing after removal of amino acid can be further means recycle or produce the other type of amino acid. For example, when if we are talking about oxidative deamination, so oxidative deamination is a process by amino group from the amino acid removed by with the help of the deaminases enzyme, and the remaining part of the amino acid convert alpha keto acid, that is alpha ketoglutarate, and this alpha ketoglutarate would convert in succinyl CoA, and this succinyl CoA can be further means convert or can be produced threonine, methionine and isoleucine valine amino acid, or maybe this succinyl CoA convert into succinate, succinate convert into fumarate, and then fumarate would be synthesized tyrosine phenylene aspartate, and so on. It can be means going in the end, the reaction will go, and they form oxaloacetate. Oxaloacetate can be form aspartate and asparagine. So anything could be happen, either amino acid um, can be means during their breakdown, they can be during their catabolism. Actually the catabolism is the process by which amino acid breakdown further breakdown, and however the anabolism is the process by which amino acid synthesize. So this is the cycle. Here it can be showing that how amino acid can be catabolized or anabolized. So for example, if the amino acid are catabolizing by the mechanism of oxidative deamination or transamination, so their metabolic product or the remaining part of the amino acid would be enter and can be recycled to synthesize or to anabolize new amino acid, or it can be completely means oxidize or completely break down to form energy after going through the TCA cycle or after entering into the glycolysis cycle. So this was whole about the um, amino acid catabolism. um, you have to just keep remember that amino acid can be catabolized by the two mechanism. One is the oxidative deamination and other is the oxidative transamination. For the what is the difference between oxidative deamination and oxidative transamination? The difference is the removal of amino group, the method by which amino group removed from the amino acid. In the oxidative deamination, what happened, the amino group from the amino acid completely lost. However, where is however uh, in oxidative transamination, the amino acid would not be lost. The amino group from the amino acid would not be lost. It would convert from standard amino acid onto the alpha keto acid, and then this alpha keto acid further recycled after going into the TCA cycle or glycolysis and then produce energy, or can be further synthesized other amino acid. The enzyme that is necessary for the oxidative deamination is the deaminases enzyme, and the enzyme that are important for the transamination are transaminases enzyme. So this was whole the about the whole amino acid catabolism, and I hope you have understand about the lecture. So for further discussion, you can go from this link, and thank you, and you can also means consult these book for further reading. Thank you very much, and Allah Hafiz.